We can solve the physics of getting to Mars. The propulsion exists, the trajectories are plotted, the hardware is manufacturable. These are engineering problems, and engineering problems have engineering solutions. What we have not solved is the biochemistry of surviving it. This is the biological audit of Mars, a rigorous data-driven assessment of what the Martian environment does to the human body upon contact. Mars is not a fixer-upper. It is a planet-sized laboratory, currently configured to dismantle human physiology. Today, we file the report. The surface atmospheric pressure on Mars averages approximately 0.6 kilopascals. For context, Earth's sea level pressure is 101.3 kilopascals. That means Mars has roughly 0.6% of the atmospheric pressure you are experiencing right now. To understand why this number is lethal, we need to establish the Armstrong limit. The Armstrong limit sits at 6.3 kilopascals. At this specific pressure threshold, the boiling point of water drops to exactly 37 degrees Celsius, human body temperature. This is not a metaphor, this is a physical law. At or below 6.3 kilopascals, the liquid water in your body has enough thermal energy to undergo a phase transition from liquid to gas. Mars, at 0.6 kilopascals, is operating at roughly 1/10 of the Armstrong limit. The planet is not close to survivable. It is not borderline. It is categorically incompatible with an unprotected human body. The physiological process this triggers is called ebullition, the spontaneous vaporization of bodily fluids under low-pressure conditions. It is important to correct a popular misconception here. The skin and the circulatory system do provide enough mechanical counterpressure to prevent blood vessels from catastrophically rupturing. You will not explode. The structural integrity of your integumentary system holds. What it does not protect are your exposed moisture-bearing membranes. The fluid on the surface of your eyes begins to boil. The moisture coating your tongue vaporizes. The liquid lining your alveoli, the microscopic air sacs that facilitate every breath you take, undergoes the same phase change. The timeline is brief. Under exposure to pressures below the Armstrong limit, useful consciousness, the cognitive capacity to take any corrective action, lasts approximately 10 to 15 seconds before cerebral anoxia terminates it. After 60 to 90 seconds of unprotected exposure, the outcome is irreversible. Mars does not present a challenge to be overcome with courage or acclimatization. It presents a hard thermodynamic constraint, and that constraint is merely the first line on our audit report. Suppose we resolve the pressure problem with a habitat and a pressure suit. We have now contained the atmosphere. What we have not contained is the soil. Martian regolith, the loose, dusty surface material covering the planet, contains perchlorates at concentrations of 0.5 to 1.0% by weight. This was first confirmed by NASA's Phoenix Nether in 2008 and subsequently verified by the Curiosity rover at Gale Crater. The Mars Odyssey orbiter has since confirmed their global distribution. Perchlorates are not a localized anomaly, they are everywhere. To understand why this matters biologically, we need to understand how the thyroid gland acquires iodine. The thyroid absorbs iodide ions from the bloodstream through a protein channel called the sodium iodide symporter, or NIS. This transporter is not selective enough to distinguish between iodide and perchlorate ions. When perchlorate is present in the bloodstream, it competes directly with iodide for access to the NIS, and it wins. Perchlorate has a significantly higher affinity for the symporter than iodide does, effectively blocking the thyroid's ability to absorb the iodine it needs. Without adequate iodine uptake, the thyroid cannot synthesize its primary hormones, T3 and T4. These are not peripheral hormones. T3 and T4 regulate basal metabolic rate, thermoregulation, cardiovascular function, bone metabolism, and neurological development. Their absence produces a cascade. Metabolic rate slows, cognitive function declines, bone marrow suppression follows, and the body begins a systemic shutdown of energy-dependent processes. Now consider the delivery mechanism. Martian dust is electrostatically charged. This gives individual dust particles an extraordinary capacity to cling to surfaces: suits, equipment, airlocks, habitat walls. Every extravehicular activity constitutes a perchlorate transfer event. Dust migrates inward. It settles on surfaces. It contaminates food preparation areas. It becomes airborne inside pressurized modules. Long-term exposure to Martian perchlorate is not a risk to be mitigated with careful hygiene. It is a mathematical certainty to be managed or not managed. To use a precise agricultural analogy, the proposal to grow food in Martian regolith is functionally equivalent to attempting to cultivate crops in a substrate laced with industrial-grade thyroid disruptors. The soil is not merely infertile, it is endocrinologically hostile. The first finding threatened your physical integrity in seconds. This one operates over months, systematically dismantling the endocrine system that keeps every other biological process functioning. Earth possesses two radiation shields that most of us have never had cause to think about: a global magnetosphere that deflects charged particles, and an atmosphere substantial enough to attenuate what gets through. Mars has neither in any meaningful capacity. It lost its global magnetosphere billions of years ago, and its 0.6 kilopascal atmosphere provides shielding equivalent to a thin blanket against a fire hose. The quantified consequence of this comes directly from NASA's Curiosity rover, which carried a radiation assessment instrument during its transit to Mars and during surface operations. The data indicate that a reference mission of 180 days in transit, 500 days on the surface, and 180 days returning would expose astronauts to a cumulative radiation dose of approximately 1 sievert, with roughly equal contributions from each phase. To place 1 sievert in context, a standard chest X-ray delivers approximately 0.1 millisieverts. 1 sievert equals 1,000 millisieverts. This mission dose is therefore equivalent in magnitude to roughly 10,000 chest X-rays distributed across the body. The European Space Agency sets a career limit of 1 sievert, a limit this single mission would exhaust entirely. The primary radiation type of concern is galactic cosmic rays, GCRs, which are high-energy atomic nuclei accelerated to near-relativistic speeds by supernovae and other astrophysical events outside our solar system. When a GCR passes through biological tissue, it deposits energy along a linear track, shredding chemical bonds, including those within DNA strands. Unlike X-rays, which produce localized, repairable damage, high-energy heavy nuclei from GCR events produce dense, clustered damage that the body's repair mechanisms struggle to address effectively. There's also a counterintuitive phenomenon called spallation that complicates shielding strategies. When a high-energy GCR nucleus strikes a dense shielding material, it does not simply stop, it shatters, producing a shower of secondary particles, neutrons, protons, and lighter nuclei that radiate outward from the impact site. In certain configurations, the secondary particle field produced by spallation can be biologically more damaging than the original primary particle. There is no shielding solution that eliminates this trade-off with currently available materials. The neurological data is particularly notable. Research on space radiation exposure has identified cognitive degradation effects, changes in synaptic density, neuroinflammation, and measurable impairment of spatial navigation and memory function in animal models under chronic radiation conditions. The animal model signal is consistent enough to register as an open and serious clinical question. There's also the light flash phenomenon. Astronauts in low Earth orbit with eyes closed in a darkened environment report seeing brief flashes of light. These are GCRs transiting directly through the retina and visual cortex, generating visual perception through direct ionization without any photon involved. This is not a curiosity. This is documentation of high-energy particles passing through the central nervous system in real time. The radiation environment is not something future astronauts will avoid. It is the background condition of the transit and the surface. The audit records it as a chronic, cumulative, and currently unresolved hazard. Mars has gravity. That is frequently cited as an advantage over long-duration zero-gravity spaceflight. The surface gravity is 3.71 meters per second squared, 0.38 g relative to Earth. This is not nothing. It is also not enough. The human skeletal system is a load-bearing architecture that maintains its structural integrity through a principle codified in Wolff's Law. Bone remodeling is driven by mechanical loading. Osteoblasts, the cells that build bone, are stimulated by physical stress on the skeletal matrix. Without adequate mechanical tension, osteoblast activity declines. Osteoclasts, the cells that resorb bone, continues, and the net result is progressive loss of bone mineral density. In full microgravity, data from astronauts document bone mineral density losses of approximately 1 to 2% per month in weight-bearing regions, particularly the femoral neck and lumbar spine. At 0.38 g, the mechanical loading on the skeleton is reduced to 38% of the Earth normal stimulus that maintains bone integrity. The losses will be attenuated compared to zero gravity, but they will occur. The loading signal is insufficient to maintain the skeletal architecture that evolved under 1G. The body does not maintain structures it perceives as unnecessarily overbuilt.
The cardiovascular architecture presents a parallel problem. On Earth, the heart pumps against a hydrostatic gradient. Blood must be driven upward against gravity to reach the brain. In reduced gravity, that gradient is reduced and the circulatory demand on the left ventricle is lower. Data from short-duration spaceflight studies show that left ventricular mass can decrease by approximately 12% even after 10 days in zero gravity, a figure that illustrates the cardiac muscle's extraordinary sensitivity to changes in mechanical loading. At 0.38 G, the gradient is partially restored, but the reduction in cardiovascular demand remains a physiological reality over long timescales. Upon return to Earth, a body adapted to 0.38 G encounters full gravity as a biomechanical crisis. Orthostatic intolerance, the inability to maintain adequate blood pressure upon standing, becomes a clinical presentation. Bone density deficits translate into fracture risk under gravitational loading. The skeleton has been progressively losing the structural capacity to handle. The body treats reduced gravity as a reason to cancel the maintenance contract on structures that no longer appear to be earning their metabolic keep. 4 independent biological systems, 4 independent mechanisms of failure, an atmosphere that vaporizes exposed tissue, a soil chemistry that dismantles the endocrine system, a radiation field that corrupts DNA and degrades neurological function, and a gravitational environment insufficient to maintain the skeletal, muscular, and cardiovascular architecture that defines a functional human body. Mars is not Earth 2.0. It is not a second option waiting to be developed. It is a hostile chemical and physical environment that is currently operating outside the tolerance parameters of human biology on every axis we have measured. The question of human colonization is not a question of willpower or vision. It is a question of evolutionary compatibility. We evolved under one specific set of atmospheric, chemical, gravitational, and radiological conditions. Mars does not meet a single one of them. The engineering is ready. The biology is not.